Hi guys, welcome back. So today we are going to talk about the chromosome. We will first talk about the chromosome structure. So you guys still remember what is chromosome, right? So chromosome is the part that contains the DNA sequence and that is located in the cell nucleus. So do you remember how many chromosomes do we have in the human cells? There are 23 pairs of the chromosome in the somatic cells. We call them diploid cells. That means we have 23 pairs chromosomes, one set from mom, one set from dad. In the sex cell, in male it's sperm, in the female it's oocytes, or we collectively call them gametes. There will be haploid, meaning that each cell only have 23 chromosomes, not 23 pairs. So in this lecture, we will cover this area. We will talk about the chromosome structure. We will get the idea that the chromosome actually not remain in that structure all the time. So there will be a situation that the chromosome is more relaxed. And in that structure of the chromosome, we call it chromatin. If it's very highly packed, it's the chromosome. But we typically call them chromosome. But with that string, we call it chromatin. We may also learn one terminology. You may know this already. If not, this will be your first time. It's called the karyotype. It's a typing, meaning that it's a way we can characterize something, but what is it? We'll learn about it. In short, it's a method to characterize the structure and the number of the chromosome in a cell. Do you want to know how many chromosomes in your cell? This will be the number. This will be the method. We say it's 423 pairs, but how do we know that? Are you sure? How do I know if it's not 23 pairs? How would I know that I am different, right? How do you know? We use the karyotype to characterize we basically count it using the karyotype, and we can also see the structure of the chromosome. And then we will learn about the structure of the chromosome, and later we will learn about something quite unique. That's the repeats of the genetic codes in the chromosome. Why is that repeat important? Is because that we typically consider the DNA sequence are the sequence to produce protein, right? DNA to RNA, RNA to protein. But in fact, that only 2% of that DNA sequence are the one to serve as a gene to produce protein. The other 98% are not. They don't produce protein. And among all these sequence, or among this non-coded sequence, 50% of those are repeats. Same thing over and over again. So we want to learn about it. So this is a typical from micro to a little bit macro, but still micro, from the DNA sequence, the chemical sequence, chemical structure into the chromosome. We learned that we know that these chromosomes are basically one single string of the DNA double strand, right? So here is a DNA sequence, GCAD, etc. Two will wind it as helix to form these DNA double strand, and then we pack it using the protein called histone right here. So these are the histone to pack it. And then when we pack it like that, this is that structure is called chromatin. If it's not folded further, we call this chromatin. But if it's further, it's like a stick like this one here. You know, it's not randomly formed like this. We need to put a lot of protein, basically kind of like glue to glue everything together. Then we call this chromosome. So this chromosome structure can be varied. When it's packed like that, packed like this, It's like glue it, right? You use your tape, you use glue to glue like that. There's no way you can read the sequence. So when the cell pack the chromosome like that, it's when the cell are not going to produce protein. And that is especially true when cells go through a cell division. So let's look at the cell division or the cell cycle. That's why we should have, and we had this cell cycle before this lecture. We get the idea what cell cycle is about, what cell division is about. If you don't remember that, please go back to my lecture. We talked about that cell cycle, cell division. So you can see that cell cycle, when it's in the interphase, these chromatins are basically freely open. It's not completely free open, but it's more open for reading. This is like a book you can read. But when the cell go into the prophase, in the beginning of the cell division, to the metaphase, when the sister chromatids are aligned with each other and ready to be pulled away into different opposite position in the cell, that is when the cells are mostly packed. And it is like this one here. So because you need to, if it's open like that, a string is like widely freely swing everywhere. If you pull it, you might tangle it somewhere and you may break it, right? So we don't want that. We want the DNA to be very fully packed like that. We put a lot of glue into it and pack it like that. And then we can pull them apart. Remember, we talked about that we are interested in how to count our chromosome, right? If the chromosomes are spread like this, it will be very difficult for us to count. So over the several years, the scientists find a way to count the chromosome. Is that if we can take a picture of the chromosome when the cells are at the metaphase, that would be the best time to capture the structure of the chromosome and are able to count the number of the chromosome. That's what we did. So this method is called karyotype. So that's the method. We basically has a cell and we just observe them. And when they are in the metaphase, we quickly take a picture and then we can see all these individual chromosomes separate from each other. And then we can count. You say that I have 23 pairs of chromosomes. Let me check. Take a picture and I count. So that's the karyotype. So two things you need to know is that the karyotype is, so the quick question is that, what is a karyotype, right? And which phase or which stage of cell you will use to conduct this method. That's right. So that's in the metaphase. So a typical one will be something like this. So you can see that you have these two, this one and this one are sister chromatids. And they are connected at their centromere. You also have a homologous chromosome, one from mom, one from dad. They are homologous, they are not identical. Sister chromatids are identical to each other. But the homologous chromosomes are similar but not identical. So that is the typical karyotype. You can see the chromosome structure. You can also see that whether you have XX, that will be a female. If you have XY, that will be a male. So that is the skill. We learn one skill, that is called the karyotype. So let's go back to a little bit. There's one more slide I did not explain. So this one. So when the cells are in the interface, like in the interface, these are not packed. And so they are formed like string, but like a folding kind of randomly, but not exactly randomly. They are still regularly folded. and we call them chromatin. So this is the chromatin. What we learned about this one is that even though they are not packed, their distribution is not overlapped, So each chromosome will spread, but spread in a regular manner, meaning that it will spread in specific territory. We call this territory a chromosome territory. So that's something we need to know. The importance about that is that these ensure that each chromosome don't really interact with other chromosomes. However, each chromosome will have interaction with the code in the same chromosome. So the sequence can interact with each other, but chromosome 1 and chromosome 2 are in the neighbors. They don't mix with each other. Yeah, so that's that. And then karyotype, we can use this to count the number of the chromosome. We can also see the structure of the chromosome. So what do you mean by structure of the chromosome? So from here, you get something that all these chromosomes look similar, but different. What are the similarity? They all have 2 arms, and their center are a little bit narrower, right? So that's what we observe, and that is what we are going to teach, right? So it's right here. So from a chromosome here, we basically have a chromosome at the metaphase. We have sister chromatin, sister chromatids. That 2 identical chromosomes attach to each other at the centromere. So from here, we kind of identify 2 very important features, that is each chromosome. This is one chromosome. This is another. And these 2 chromosomes are one chromosome, the other chromosome, and each chromosome has 2 arms. The shorter arm, we call it a P arm. P refers to the petite, meaning it's short, and the Q arm. Their centromere, the end of each chromosome, we call it telomere. So that's some feature. From here, we also see that even though it's like a stick, right? It's a centromere, it's a bit narrower, a stick, right? But the density of that DNA in that region are different. We see some regions are more compact, some regions are more loosened up. So this compact region, we call them heterochromatin. The more open region, we call them euchromatin. And in the heterochromatin, why is that more condensed? Yeah, it's more condensed. So it's a DNA sequence in that region, even though if we open up, that region is tend to be more condensed. It's a region that the code is not revealed, it's not exposed. So those codes are not for protein synthesis. And so in the heterochromatin, and in contrast, the euchromatin is a region that is, even though it's packed, but it's more loosely packed. So it can be opened, and those codes are for the protein synthesis. So it's called the coded gene, and these are called the non-coded DNA, and this is for the content coded DNA. We mentioned that there are repeats, right? 50% of the codes are repeats. Those 50% of codes are usually occur in the heterochromatin. So in this region, if you do really open it, even though it's packed, if you do really open it, you will find that has a lot of repeats. For example, if this is code, right? Code means that it's code, and so it provides meaningful information. And so you will be curious that, okay, what's our ancestor trying to tell me? So I open it up, right? It's a book, right? And then you find that in this region, it right, dear my grandson, you're supposed to be a good man, right? You are a good man, something like that. So this code provides a meaningful sequence. And if when you read something meaningful sequence, it makes sense, right? But in this region, and it's packed, it's very difficult to open. But when you open, you find that there are a lot of repeats. You find it will be like like repeats of I, or repeats of say what's that? For example, under say Mary, Mary, M-A-R-Y, right? Repeats. So when you read that, you realize there is a glitch, right? There is a computer glitch that is not an article, and you continue you read to this area. Oh, it makes sense. Dear my grandson, something like that. Yeah. So this heterochromatin, it's more packed, and it's a protection, right? So it doesn't want we to read it, or it doesn't want the transcriptional machinery to read it, because it's not for them to read. It's packed. And there are several theories. Why do we need to store about 50% of repeats in our code? One possibility, one hypothesis is that they are not for reading, they are for the connection. So here is we see this heterochromatin particularly condensed in the telomere and the centromere. So in the centromere, this region that we allow two chromatids to connect with each other. So they provide that connection. They don't really are the purpose to be read. As for a telomere, you know that when the DNA replicate itself, they cannot replicate the very end. So every time when cell divide, the telomere will be shortened and shortened. And you will be worried that if that are contain important code, right, then you lose that information. So in order to prevent that, what nature does is that just put a lot of repeats in the end. So even though it gets shorter and shorter, it doesn't really matter, because that's the code we don't really care. Something like that. It's just hypothesis. So the difference between the chromatin and the heterochromatins are more compact, and they provide non-informative information. They don't provide informative information. In other words, the code there contains a lot of repeats. So that's that. And in contrast, the euchromatins are more openly accessible, and their codes, they contain the genetic codes for protein synthesis. All right, so that's that. So from here, we learn the chromosome structure can be dynamic. We want to count the chromosome number. We will wait until cells are in the metaphase. And at that moment, we take a picture under a microscope. Then we can get the Photoshop to put chromosome side by side from the largest one to the shortest one. And then we can count. In the normal cell, the somatic cell, we're supposed to count 23 pairs. From there, we can also identify the person's sex, whether it's XX or XY. From each chromosome, we can identify short arm and the long arm. We can also identify regions that is heterochromatin or euchromatin. And you should know that which one is the coded, protein coded region that is euchromatin. You will see this in a quiz question though. Yeah, I guarantee you. Not guarantee, I don't know. But it will be likely that you see question like that. Okay, so this is an example of the telomere. We say that telomere contains a lot of heterochromatin, so it's highly compact. They are not supposed to be read, but if you do open it, try to see what's there, you see a lot of repeats. In the telomere, for example, you see repeats like TTAGGG. Here it shows you 2 repeats, TTAGGG, TTAGGG, but in fact, there are hundreds of thousands of that repeats in the telomere. And so every time you divide the cell, telomere gets shortened and shortened and shortened, but it doesn't really matter because those repeats are not supposed to be, it's not going to be transcribed anyway. Yeah. So that's the importance of the repeats, importance of the heterochromatin. It's more like structure, but they are more than that. Structure is just one possible function of those codes. All right, so we know that structure, right? We have the P arm and the short arm. And we can compare the length of P arm and short arm to classify the chromosome into one of these 4 types. If the P arm and the short arm has equal length or similarly equal length, we call this chromosome a metacentric chromosome. If the P arm is shorter, but is substantially is still there, we call it submetacentric chromosome. If the P arm is much shorter, then we call it acrocentric chromosome, like this one here. It's much shorter. It's still there, but it's very short. And if the P arm is barely visible, we call them telocentric chromosome. So among all these 4 types, you may see quiz question like which one is the metacentric chromosome, like P equal to Q or P much shorter than Q, etc. But among these 4 different chromosomes, different type of chromosome, one type I want you to pay a little bit more attention is the acrocentric chromosome, this one. P is much smaller than Q, like this one right here, this one here. Acrocentric chromosome. It's because there are 5 somatic autosomes, like non-sex chromosomes are acrocentric chromosome. I would suggest you remember this number: 13, 14, 15, 21, 22. These 5 are acrocentric chromosome. Their P arm is much shorter than the Q arm. And one thing about, there are several unique things about this acrocentric, this 5 chromosome, which is that this P arm contains heterochromatins, meaning that it contains repeats and it's highly packed. That may not be too weird, but another thing is unique, especially unique about this acrocentric chromosome is that these P arms, they are repeats, they are highly compact. And another thing is that they have identical repeats. So the same repeats in 13P are in 14P, 15P, 21P, 22P. They have the same repeats. These repeats, there are several repeats, but they spread in these 5 P arms. And these repeats produce. It's not the one that don't produce. They don't produce protein, but they produce the ribosomal rRNA. And there are some region produce protein too. So that's the unique part. These P arm in these 5 chromosomes. So let's take a look at these 5 chromosomes. See how small their P are. 13, 14, 15, 21, 22. You can see that their P is, we call this P arm the satellites because they kind of hang out there. Satellite chromosome. So So that's this one here. Satellite chromosome in these 5 eccentric chromosome. They contain the codes, they contain the same repeats. These repeats are the codes for the ribosomal RNA. And that repeats not just one copy, they have multiple copies. So that's how we produce so many ribosomal RNA out there to form the ribosome and to conduct the translation. So we have many ribosomes, right? Let me show you the picture like this one here. So this is the nucleus. All these little dots are ribosomes. So we synthesize a lot of ribosomes. And that's because we have many copies of this ribosomal DNA code spread in these 5p arm of eccentric chromosome. Yeah. So this p arm contains these chromosome segments kind of hang out there. We call them satellite chromosome. They are connected with the centromere through this string. We call them stalk. And these satellite chromosome or SAT chromosomes contains the codes to synthesize ribosome. These heterochromatin are also, when they are in the interface, each chromosome has its own chromatin territory, right? Chromatin territory, and this p arm will located in similar area. And that area is called the nucleolus. So let me show you nucleolus. So this is like this here. So here in the interface, different chromosomes are in different territory. Some regions are more condensed. We call them heterochromatin. This one here, that's a heterochromatin of each chromosome in their territory. But you can see that these heterochromatin are especially large. That is because that's a combination of the p arm from these 5 chromosomes. So these heterochromatin just gather in this region. Yeah, even though they are spread out, yeah, in this region. This is the region of this region is called the nucleolus. Nucleolus is a region inside of a nucleus. You should know this cell already. This nucleus contains the chromosome, and this is the nucleolus. This nucleolus contains these 5 p arm from eccentric chromosome 13, 14, 15, 21, 22, yes. And then their function is to synthesize ribosome. So that's that. I don't think I missed anything. Good. We can also use these from here, we can also further to use this to give the address of each individual genetic sequence. So for example, that basically we have a chromosome 1 to 21, sex chromosome X, sex chromosome Y, and we have P arm, Q arm, and each arm we can also segment it into 1, 2, 3, 4, etc. And for example, that a gene called the CFTR here, just for example, and this is a gene for the coding for the cystic fibrosis transmembrane conductance regulator. So these gene codes are located in, for example, in this region. The human is in the 7q31.2. So from here we know that it's in the chromosome 7. So each chromosome has this number. Like this one here, 1, 2, 3, 4, 5, 7. So it's on the chromosome 7. It's P arm, Q arm. So this code is in the Q arm, right? In the Q arm. Then Q arm, we can have one from the centromere to the telomere. We can have 1, 2, 3. So it's in the 3 region, one, maybe 1 and 2 and 3 region, and in the 3 segment is either in the 1 or 2, it's in the 1, and in the 1 is in the 1, 2. So it's 7q31.2. So we can give the address to each gene. We call them the location of the gene, or we call it a locus of the of a gene. And that coding method is called the ideogram to code gene based on its location in the chromosome. We can code it in the chromosome in this metaphase structure, or we can still do the same thing in the interphase structure. Here you can see that this is the interphase, so it's not packed like a stick, but more like spread away. This is a chromatin. And you can still see that some of these regions are highly condensed, especially in the telomere and the centromere. There's still also some heterochromatin, like compact area or repeats area in this region. So as I mentioned earlier, about 50% of the codes are repeats in the sequence. So that's that. So talking about the repeats, let me drink water very quickly. So talking about repeats, right? There are 50% of the codes are repeats. Let's learn about the repeats. So what are the repeats? The repeats are repeat sequence in the repeat codes in the sequence. As I mentioned earlier, in the telomere, right? Let's use the telomere as an example. We learned that it's basically the same code, TTAGGG, over and over. So that is a repeat. The unit of that repeat has 1, 2, 3, 5, 4, 6, 6 code, 6 bit base pair, 6 base pair, right? 6 base pair. And so that's 6. And that unit repeats and repeats. The repeats can be classified into 2 major types. One is a continuous repeat, like this one here, TTAGG. There is nothing between. There is no space between. And there is another type of repeat, it's a TTAGG, and some other sequence, and then TTAGG, and then TTAGG. They are repeats like that. So the repeats will be here, and then there, and then there. Yeah. So there are 2 types of repeats. So go back here. About half of the human genome. Genome means the code of entire organism, like a cell. Entire code is called genome. A half of that entire code are repeats. 2 types of repeat. The continuous repeat, we call them tandem repeats. Non-continuous, we call them interspersed repeats. So let's learn about these 2. They are very common in our sequence. They don't really provide functions. Some of them provide a function, but majority of them don't. So the unit is that it can be 1 base pair, for example, a very like I or T, something like that. 1, or it could be 6, like what we saw in the telomere, or it could be several thousand base pair long, and then it repeat and then repeat. And the length of the repeat means that it has 1 copy or 2 copy or 100 copy or 1,000 copy can be varied. Based on the size of the repeats, like the length of each unit, if the repeat units range from 10 to 6, like 10 codes to 60 codes, is called a minisatellite repeat. We see these repeats more common in the centromere. If the base pair are less than 10, like in the telomere, like the one in telomere, still remember that? I don't remember. What's that? TTAGGG. TTAGGG. So that's 6 code, right? So this kind of repeats are called the microsatellite. So basically, it's kind of like satellite, like remote area. It has multiple repeats. Yeah. So that's like that, mini satellite or microsatellite. So let's take a look at it. So tandem repeats is continuous. It can be mononucleotide, for example, multiple A, dinucleotide, CA. There's common ones, CG. In the future, you may see that. It's called the CG island. And then it's a trinucleotide, 3 codes, 4 codes, 5 codes, 6 codes. Yeah. So that's that. So it repeats over and over again. These are the tandem repeats. We also have a interspersed repeats that code repeats but not continuously. This is an example that's imperfect but nearly close. It's because that even though it's a lot of these interspersed repeats, over time there will be mutation. So one of the codes will be buried. Yeah, but you know, by comparison, we kind of get the idea that they are supposed to be the same, but over time one code become changed, and then this is So for example, here we have a ATGG TTC, ATGG TTC. So that basically is a repeat. Now we have another one, ATTC. So that is also repeat. This TTC, maybe many years ago, many 100,000 years ago, they are the same. But over time, this one changed or this one changed. So that is just an example of the interspersed repeats. So what do we learn from here? We learned from that 50% of codes are repeats. We see a lot of repeats in centromere, telomere. These repeats, if they are open, they can generate codes, right? And that they are not meaningful codes, so we don't want them to generate to be read by our system. So we pack them very tightly, and that leads to the term heterochromatin, that is condensed chromosome. So that's that. One thing we also can use this, even though they are not codes to provide protein synthesis. We can use it in very interesting way. Yes, we can use it for a very interesting way. Let me think. What can we use about that? So they are repeats, right? Repeats. But we don't read them. They don't produce protein. Then you say that we can use it for what? Okay, let me show how we can use it. Turns out that these repeats, they don't produce protein, so they don't actually affect our body function because they don't synthesize protein. But these repeats are there. And in other words, the copy number, 1 repeat, 2 repeat, 3 repeat, the copy number doesn't really matter because they are not going to generate codes or generate any function. Doesn't really matter. So people can have different number, even though we have the repeats, but the copy number are different. Someone we have 2 repeats, some people have 6 repeats, some people have 100 repeats. For example, just for example, people don't really have any disease from that. So this lead to the idea that variable number random repeats. So that's the idea that the number of the random repeats can be varied. These repeats have a microsatellite or minisatellite. One of those is called the short tandem repeats. So let's look at this one. So let's read this one very quickly. So the nuclear sequence of the repeats keep in our body. And so my mom to me, my dad to me, my dad may have 6 repeats, my mom may have 3 repeats. It doesn't affect their function. I will get 1 copy from my mom, 1 copy from my dad. So in my 2 chromosomes, I will have 1 with 6 repeats, 1 with 3 repeats, something like that. So the number can be varied. And these repeats, one very unique one is short repeats, right? The less than 10. For example, short tandem repeats, short for STR. Let's look at what is STR. For example, like this one here, short tandem repeats. Here, the best units are 6 bits. Short tandem repeats tend to be 2 to 6 bits base pair, not bits, base pair, BP, base pair. And so we have 4 codes, and then again and again and again. Your mom may have 4 repeats. Your dad may have 6 repeats. And then you will get 1 from your mom, 1 from your dad. So your 2 chromosomes in the same location may have 4 repeats here and 5 and 6 repeats there. So each individual will have its codes based on the number of repeats. These repeats will be kept because nobody would die from it. They don't really generate any protein. So nobody will get sick by having less repeats or more repeats. So those codes will be kept in you. And so that codes indicate you're relevant to your mom or your sister, your brother, your siblings, your aunts, your relatives. For example, let's see this one here. Now we have identified 13 regions. These 13 regions contains STR, short tandem repeats, STR. These 13 regions, short tandem repeats. Let's just give an example. Your mom and your dad has 2 repeats for every repeat. And you will have similarly 2 repeats in every repeat. However, your neighbor will be different. Your neighbor will be 10 repeats for every repeat, or 5 repeats of these repeats, or 2 repeats here. And so each individual will have its repeats variable, and closer your biological blood relates to, the similar your repeat variable will be to each other. So that's that. And so we can use these to identify whether this is your son or your daughter, right? If their repeats is very further away from you, then you know that this might not be my kids, right? Something like that. Another thing is that we can use this to identify the remains, say, when in the war, right? If the body are destroyed, we cannot identify whether these legs or this body belong to this soldier or etc. We can get the sample, we can see their repeat variable, and to match it with the sample of that family, right? And so we can identify the body. Yeah. So that is the 13 short tandem repeat markers used to identify individual. The FBI use combined DNA index system. And if the person, it will be very, very rare, 1 in 250 trillion chance to have 2 people have identical number variables of that repeat. So I think that's it. Yes, so that's that. All right.